Hi everyone. I'm Vincent Cervantes. I'm a second year postdoc here in UCR. And welcome. Thank you for joining us to this webinar that is part of our inclusion subcommittee in the uh, North American Arabidopsis steering subcommittee. Uh, so we're gonna do this with a little introduction of the panelists, which is Stefan DeFolter, Dr. Nayelli March Martinez, and Dr. Stewart Gillmor, all of them from the Center of Investigation Advanced Studies of the National Polytechnic Institute and CINVESTAV-Irapuato. So the structure of the webinar is gonna be like first the welcome. I'm gonna give you this program outline. So make sure that your audio is muted. Just remember that we're gonna record this, and well, then we're gonna have the introduction by the panelists. Each of them are gonna talk are gonna give you like a brief introduction of 5 or 10 minutes, and then we're gonna ask some base questions, and then we're gonna have time for some submitted questions that you can start submitting in the chat. So if you want to start doing that after the panelists they do their introduction, you can do that. And just a reminder that we are a community which is open to all in North America. So if you're from Mexico, from United States or Canada, so you can join. You can become a member to the NAASC Arabidopsis community group. So the membership, which is annual, it starts to $10. So you can just copy this, or if you are interested in joining to this community, which is really good, where you're gonna find a lot of opportunities. So you can just join us, please. So we're gonna start this with the introductions. So we have first Nayelli March from CINVESTAV-Irapuato. She's gonna give us a little introduction. Thank you, Nayelli, for joining us.
Thanks a lot, Vincent, for the invitation. Thanks, Joanna, also to help us to connect and all the organization, and for the NAASC to provide this space to talk about our story. Well, so I'm Nayelli March Martinez. Thanks a lot for connecting, for taking a little bit of your time to listen to us. I work at CINVESTAV-Irapuato in Mexico at the Department of Biotechnology and Biochemistry. So we were asked to answer 5 questions, these questions. I'm going to answer them one by one. But first, I want to tell you a little bit about my path. So I'm Mexican. I started to study my PhD in Mexico. And then when I was starting, my boss asked me if I would go to Holland to do a collaboration project. So I was very happy. I went to Holland. I was supposed to be there 3 months, but the 3 months became 3 years. I met my husband there. And then several years later, I came back with him and with the children. So one went to Holland and 4 came back to Mexico. So I studied biochemistry engineering as a bachelor. That's my bachelor degree. And then I went on to study a PhD in plant biotechnology at CINVESTAV-Irapuato here in Mexico. So I did this PhD with Luis Herrera Estrella and also with Andy Pereira. So I did most of my work in Wageningen. You are in the Netherlands with Andy Pereira. Well, this was like sort of a co-direction. And my work was about building an activation tagging population in Arabidopsis using transposons. Later, at that moment, I met Stefan, who is my husband. He is now here in Mexico. I was then a postdoc at the group of Andy Pereira, and then our first son was born. Then I had to take a pause, a childcare pause, because what I was going to earn as a postdoc was less than I was going to have to pay for childcare at that moment in the Netherlands. So I decided that it was better to take this post. No? And Stefan was finishing his PhD, so it was not very easy to balance everything. And then I went on to do a second postdoc with Gerco Angenet in the Netherlands and Elena Alvarez Gulia at UNAM. And at this moment, our first daughter was born. our daughter was born, Elisa. And then we came back, I came back, and they all came to Mexico, where I worked first as lab staff at the Advanced Genomics Unit at CINVESTAV, then as a visiting researcher at a different department, and finally I was hired as a professor researcher at the biotechnology and biochemistry department at CINVESTAV-Irapuato. So that's more or less my path. And well, the questions, no? So what inspired me to dedicate to plant sciences? Actually, it was very hard for me to decide. I love many things. I even started a career in international commerce, but I didn't feel that it really made me so happy. So I studied biochemistry, and then I always liked plants, and I realized that they are essential for our life, no? For the environment where we live on, for the food that we eat, the materials that we use, even for energy in the past and in the future, I hope, for many medicines and many daily stimulants that we use. No, so they are essential. They are also beautiful. They are very important, I think, for our mental health. And once I went actually to an open day at CINVESTAV, and then I saw a Petri dish with callus, and that just made me crazy about them. And I said, no, I have to study plants. No, this plasticity, this wonderful way they have to regenerate themselves. I love it. So I decided to pursue the PhD in plant biotechnology. And then the next question was, what challenges have you faced? Well, I think we all have faced a lot of challenges. So maybe, well, at the beginning, being a Mexican woman in Europe, it was a little bit complicated because the culture is very different. No? So I had this cultural shock. Later, I adapted and I liked it a lot. I also was supposed to be there 3 months, but things were going so well that both bosses were telling me, no, you should stay longer. But well, I only had a fellowship that was like the Mexican fellowship for Mexico. So the money was, of course, not enough. So they helped me, and I had to get extra jobs to remain in Holland. But also I was super stressed because I had to finish every time things in 3 months, and then 3 months, and then 3 months. So by the time I finished my PhD, I was totally exhausted. I even said, I don't want to put a foot in a lab anymore. But well, at the end, I realized that actually it's very cool, so I came back. No, something that happened when I was in Holland was that when I came, the anti-GMO movement had not started. So GMOs were still seen as very cool. There were products sold in the market, like this flavor saver tomato. And then I remember I went on holidays to Mexico, and when I came back, suddenly there was this boom, like people don't want transgenics anymore. They started to destroy trial fields and so. And then the whole plant field, at least in Europe and in the institution where I was, started to get to be less regarded as bad, no? So there was less financing, and things went not very well, not as they were, no? Another challenge that I have had is, I had a lot of self-doubt. When I went to Holland, I was thinking, no, I'm not going to make it. I cannot do it. They are great there, and I'm just a Mexican. No? And later you learn that, no, actually, you can do it. We are all people. You can find all kinds of people everywhere. I guess I still have something like that. I still have sometimes this imposter syndrome, and I have learned that this is very important. Not important. It's very, very common. Many people have it. No, so you have to learn just to deal with it. Do your best. Another big challenge, I think, is when I became a mother, it was like I felt like people thought that my brain had drive. No, it was really amazing. So as a woman, I felt some discrimination, but not so much. But when I felt it really hard was as a mother. No. And well, the other thing, of course, is all the failures that you have. My first paper was rejected. My first project was not granted. All these things that you have to, that you will be rejected, and you just have to keep on working. We also had very hard family health problems, and this made me also sometimes think about quitting. Fortunately, things became better, but yeah, it was quite hard. And well, there's the financial side as a student, and now as a PI, you have to find resources. And I think more recently, a big challenge is the bureaucracy that we have in Mexico and probably in the world. Also internally, it's becoming harder to do things sometimes. Then the next question, was which path did you take? Well, I started to do yoga and meditation, and well, I just kept on trying things. If my paper was rejected, I tried to improve it and resend it. I had also to decide how they say, you have to choose your battles. So sometimes things were just not worth following, and then you had to let them go. And the rest, the things that you really care about, you just keep on going. For example, when I was taking care of my first baby, I was also writing papers. All my friends were working in the lab. My family was not there. The one of Stefan was very far, so I had to take alone care of my baby, and it was really hard to write a paper and take care of it. So I don't advise it, but if you have to do it, you do it. And as Jose said, actually, many of my decisions were based on what was best for my family. So what I did is I just tried to do my best, knowing that my best one day was going to be different than my best from another day, It's just like that, and don't beat yourself in one day you just cannot move. No, it's not like that. No, but yes, you have also to be gentle with yourself. I'm also now trying to participate more in outreach activities because I saw how destructive was that as scientists, we did not get involved more in educating the public about what we were doing with the transgenic part. Then the next question was, who helped you along the way? Actually, many people. I feel super grateful to many people. So, well, my family, of course, my parents and siblings, my mentors, Andy Pereira and Luis Herrera Estrella, they really gave me a golden opportunity to move to learn in Holland. It was really it changed my life, so I'm super thankful for this. And I met super interesting people there. I met Stefan, and well, from Andy Pereira, I learned a lot about transposons and molecular biology, and from Luis Herrera, I learned a lot of how to do science, and also he gave me a lot of freedom. I'm very thankful for what I have learned from them. Then, well, I had many friends in Holland and in Mexico that helped me to keep going, that provided a very warm and super fun environment, and many of them were in the lab, so even in technical issues they could help me. I'm also grateful for them. I could not put all the names and pictures because it will be too much. Of course, well, Stefan, I put the picture because you are going to see him in a moment. He was a super great support. He was very understanding when I was super stressed when finishing the PhD. I'm very thankful for him. And now we are still great, partners, I would say, in life and even at work also. Also, for my postdocs, I work with Herco Angenen and Elena Alvarez, and I also thank them for the opportunity to go back to science and for the freedom that they gave me. And I feel super grateful to my colleagues in the biotechnology, biochemistry department, because they believed in me and gave me the opportunity to work as a researcher with them. And the institution where I work, I have to say, it's not perfect, but I have felt very supported, especially in the very hard times of the family health issues. They have been, in general, very understanding, as well as my colleagues. I have felt supported by them. They have been super understanding also. And actually also, like the people that work at CINVESTAV, even if we have a very tight and terrible bureaucracy, they always try their best to help us to keep on working. So I'm also grateful for them. And well, at the end, my students and my children, this is when they were small. Now they are teenagers, and I don't know if they want me to take their pictures. So actually, they keep me inspired. No, I really like to see how they develop. These young people, they develop their minds, their skills. It's really an honor to see the developing. Sorry, and my children also. No, I have now to almost let them go, and it's not easy, but I'm very proud of all them. And finally, what lessons have I learned? Well, first, don't leave things to the last minute. No, that have cost us one project to Stefan and me once. It was not easy to balance children and work, and no, but yes, don't leave things to the last minute. I have learned that many things are in your hands, but many things are not. No, and sometimes it's hard to distinguish, and you get angry because of things that actually you cannot do anything about, and some things you can do, and then well, the best is just to do. I think I sometimes you have to do things because you have to do them, and then you just do. But I see that the best is when you do things because you love them. So you love plants, and then you work with plants, and yeah, you don't know where this will bring you. But if you do it with love, most of the times things are turn up quite well. So I have found that in this path. And sometimes yes, you have to leave things, to let things go. And like Jose said, well, first also thanks, Jose. His talk was amazing. I think it was he's super wise. I agree. Don't care too much about what people think about you. Sometimes they will have an impression of you, but sometimes it's not what they think what matters. It's what you think and what you want to do and what you love to do, and no. And also, there will always be challenges. Don't put your happiness on when I have this, or when I finish that, or when I get the paper accepted, or then I will be happy. Because you get the paper accepted, and then you go for the next, or you get the position, and now you have a lot of work. There will always be challenges. Don't delay your happiness until you get to what you think is your aim. I have found that enjoying the small things every day, a smile, a super cool paper, a hug, even a chocolate, a nice coffee, a flower, those are, I think, things that help you to recharge and keep you going. And well, that was my presentation. I hope I didn't make too much time. Thanks.
Thank you, Nayeli. It was a beautiful introduction, and you actually answered like all of the questions that we had, and thank you so much for that. And yeah, like inspiration was like one of the main questions, and it was really beautiful. Thank you for that introduction. So we are gonna continue with Stuart. So if you can like start sharing. Thank you, Stuart, for joining us.
Yeah, so first of all, thanks for Vincent for inviting me. It's fun to do something like this. Don't think I've ever given a presentation exactly like this before. Thanks to Nayeli and Stefan for participating also. Nayeli, very interesting presentation. I think everybody has different challenges along the way. And I think Nayeli was lots of very interesting things to say about the challenges that women and Mexican scientists face, especially. So yeah, so I'm Stewart Gillmor. I'm from the United States. I'm from Connecticut, which is the southernmost state of the New England region of Connecticut, which was where the first colonies were. I'm actually on vacation now at my parents' very, very old house here in Connecticut. This is my lab website, in case you're curious about what we do in my lab. As far as what inspired me to study plant science, this is a picture of my parents' backyard and their garden and the forest, and this is where I grew up. And so I was pulling weeds from a very early age and doing gardening and moving wood around, and really, I grew up in nature. And I think that's a lot of what inspired me to study plant biology. When I started university, I went to a small college, so only about 1,000 students, and I was originally going to study either math or philosophy, and then I decided that those weren't right for me, and then I was maybe going to be pre-med, and then I took an introductory biology course, and I really liked the section on plant biology, plant physiology, which is what we primarily learned back then. And it's a picture of my sister and my parents there in the corner. They also inspired me. My mother is a scientist. My father is a historian of scientists, and my sister ended up being a medical doctor. So we were definitely encouraged to do science stuff growing up. So what I kind of grouped together what path I took and what challenges that I faced. One thing that I always recognize is that I'm very fortunate and I've been very privileged for most of my career. So I did my bachelor's degree in biology at Swarthmore College, which is a small like university that we have in the United States. They're called colleges. They typically don't have graduate students, so the professors spend all their time teaching students. And I had a couple of influential professors there, like Mark Jacobs, who was a plant physiologist who did very important early work on auxin, actually. And after I got my bachelor's degree, I thought that I had this notion that I wanted to be a professor, but I didn't feel that I was really ready to go to graduate school. And so I got a job as a research associate at Rockefeller University in Nam Hai Chua's lab, which is a very large plant molecular biology laboratory. So I was clueless when I got there about how a big lab worked, but I really learned a lot there. My supervisor, Dan Chevelle. And then I did my PhD at Stanford in Chris Somerville's lab, which was actually located on the Carnegie Institution grounds, which was a plant biology institute on the Stanford campus. That was a really great time, all kinds of exciting stuff going on at Stanford at the time, like the first microarray was invented around there at that time, all kinds of really exciting research. And after I finished my PhD, which was on Arabidopsis cell biology, genetics, basically studying mutations that cause embryos to have different morphologies, because I was really interested in studying how cell elongation and cell shape is controlled. I did a short postdoc at CIMMYT at the International Maize and Wheat Improvement Center, CIMMYT in its acronym in Spanish, in Texcoco. I was working with the Apomixis group at the time on reproductive development in maize. And then after a year or so at CIMMYT, I started a postdoc with Scott Pothig at the University of Pennsylvania, because after learning genetics and cell biology, and in my PhD, I wanted to learn more about developmental biology. And so Scott Pothig is a very prominent developmental biologist, and he mostly works on a regulation of leaf traits and vegetative phase change, but he also had a project on embryogenesis, so I started working on pattern formation in embryos in my postdoc that was in 2004. And so yeah, and then I when I was looking for jobs, this great opportunity came up at Lankebio, as it was called at the time. Now it's known as the Genomics Unit of CINVESTAV-Irapuato. I started there in 2010. I've had many excellent colleagues there, students and postdocs and staff. And in terms of challenges that I faced, like I said, I really feel that I've been very privileged. Most of my education, my main challenge was just my own limitations, trying to overcome my own limitations to do the best that I can. I was always surrounded by people that are very brilliant, much more talented than me. And so I always say it's easy to be humble when you know so many people that are so smart and talented. So that was my privilege. That was my main challenge, being supported by really smart and energetic people. And it was like that also when I started in 2010 at the Lankebio. We were very fortunate. Like I said, excellent colleagues. I've had excellent students and postdocs. Starting at around the pandemic, or maybe a little bit before, that's where I faced my greatest challenges. Obviously, the pandemic was a challenge for everyone. I've noticed that there's a lot of apathy towards science, towards really doing your best starting in the pandemic. I think it's a worldwide phenomenon. Um, all of us in Mexico now, we're in a very challenging research environment, especially in terms of getting funds for research and just, I think, a general decrease in interest in science. So that's I would say my greatest challenge currently, just trying to run a lab in the current environment. Um, so who've helped me along the way? Three very important people who've been there almost since the beginning. My wife, Maria de la Luz Gutierrez Nava, who is doing a PhD also, who I met at Stanford. My buddy Sean Cutler, who started his PhD at the same time as me in the same lab. So we were there the whole time. And my mentor during my PhD, Wolfgang Lukowitz, who was a postdoc, who started maybe a year and a half or something after I started. And so all three of them have been a great source of inspiration and learning for me from starting really a long time ago now and continuing. So I'm really grateful to have them as partners and friends. And of course, there's a long list of people that helped me in different stages of my career. Chris Somerville was a really awesome PhD advisor. I really could not have asked for anything more. We had lots of resources and lots of really smart and talented people. It was an amazing place to do a PhD. I remember when I started, Chris told me that he was interested in why Arabidopsis was 30 centimeters tall and an oak tree was 30 meters tall, and anything that answered that kind of question would be fine for me to work on in my PhD. So I always really had a lot of freedom and resources. I had a lot of great colleagues, either postdocs or other professors or postdocs or students during my PhD. I did a short postdoc at CIMMYT, as I mentioned, with Danielle Griminelli and Olivier Leblanc, and that's also where I met Daphne Autran. They're colleagues that work on apomixis and reproductive biology. And then in my postdoc with Scott Pothig, I really, really learned a lot about developmental biology and about how to plan experiments and all kinds of things. I'm very grateful for that. And with other colleagues there at Penn, other professors or other lab members who were source of support and knowledge for various different things. And then also starting since I started my lab at CINVESTAV, I've been very fortunate to have multiple different colleagues and collaborators who contributed in one way or another. Luis Herrera, thanks for hiring me in the first place. I appreciate that. And I've had, many, many great collaborations. It's something that I really, really appreciate. It's really helped with whatever success that I've had to have so many great collaborators around. Let's see here. I'm almost at the end. And of course, my lab members, I can't thank them enough. I've had, or have, I guess maybe more than about 30 students and postdocs at this point. They all made contributions to the various projects we have ongoing. And that's I think one of my greatest satisfactions being a mentor and trying to help people realize their potential and do something interesting with their lives. As I always tell people, that's one of the most important things to figure out is what is a really good thing for you to do. And for everyone, it's a different answer. You don't want to do something too difficult, or also you don't want to do something too easy. And there's all kinds of different things to take into account. So I always try and help everyone realize their goals. Thanks also to Jessica Calcagno, my lab manager, who has also been there almost since the beginning, since I started at CINVESTAV. So she's been a great support over the years. And so as lessons that I've learned, I think it's important to find, if you're lucky enough to be a scientist, we are very lucky because we can do something that's so interesting. And in science, it's really important to find what you love. So for me, what I love is Arabidopsis genetics and embryology and genomics. I started on that path almost 30 years ago now, and it took me a little while to figure out what I was good at, but I finally did, and that's been really enjoying it a lot. Some other advice or things that I've learned, it's really important to pick a good question. And probably you want to stick with it. Not necessarily, maybe you decide that it's no longer a good question eventually, but if you pick a good question from the beginning, it's probably going to continue as a good question for many years. Something else that I think it's important is it's really important to be an expert in something. You don't need to be necessarily an expert in everything, but you really do want to be an expert in at least one thing. Otherwise, honestly, you're not that useful. So multidisciplinary stuff is good. And if you can be an expert in more than one thing, that's fantastic. But you should at least be an expert in one thing. And another thing that I've learned is that you ask other experts for advice. You can get so much from getting advice from other people who really know what they're talking about. So that's something that's really helped me a lot. Read the literature, something that's super important. And when you're doing lab work, read the protocol and try and understand what's going on in the protocol that you're doing. I think this is, that's it for my presentation. Thanks a lot for listening. Be happy to take questions at the end.
Thank you so much, Stuart. It was a great presentation, very inspiring. Thank you so much for joining us and accepting this invitation. We are happy to have learned more about you. Thank you so much. And then we have Dr. Stefan DeFolter. Thank you so much, Stefan, for joining us.
Okay, so my name is Stefan DeFolter, as you heard from Nayelli in the first talk. So my story. So here's a picture of me quite some years ago. More hair, a bit younger. This was when I just arrived in Mexico, 2007, when I started my lab. And so originally I'm from the Netherlands, so I'm Dutch. I did all my studies there, undergraduate studies and graduate studies. Mainly I worked in Plant Research International. I got my PhD at the end from the Radboud University in Nijmegen. At that time, let's say Plant Research International is a research institute which is now fused with the Wageningen University. And at that time, my boss became 1 day a week professor in Nijmegen. So I only went to Nijmegen to defend my PhD, but all the rest I did actually in Wageningen. And my undergraduate studies was in plant biotechnology. The same actually is one of big or main field what we have here in Irapuato in CINVESTAV. Then actually, so I did my undergraduate and then I started to work as a research assistant with the idea to do this a few months and then travel. I bought my backpack. I really wanted to go to Australia and then travel. And then every time they asked, do you not want to stay a few months more? And with that, after a few of those times that my contract got renovated, I got a permanent contract. And then actually I stayed. I really liked the science. And at a certain moment, actually, I started to do a PhD, which was not my original plan. So I will come back in a few slides to that. But I'm very happy that I did this. So I finished in 2006. Did a short stay at that moment also of 2 months in Mexico. And like 1 year a postdoc still in the Netherlands. In 2005, I did a presentation about my work actually here at CINVESTAV. That's how it started all a little bit and why I ended up here also. And after 2007, so when we moved, so Nayelli already did a very nice introduction. So we moved with the whole family to Mexico. Nayelli seriously asked me if I was really sure that I wanted this, and we had actually quite a good life in the Netherlands. But well, that was for me the adventure also to actually leave the place and then be able to start our own lab. So well, I think actually my career went quite successful. I got two national prizes here in Mexico, and at the moment I'm a full professor here at CINVESTAV, and recently also actually became editor-in-chief of a Springer journal, which is called Planta. So here's some text, so it says more in detail. So what I did, so I mentioned already my undergraduate studies. It was with Mark Aerts, directly starting with Arabidopsis. Before the Arabidopsis genome was available, cloning genes, making transgenic plants, overexpression lines, co-expression lines, and we'll do a lot of histology and all you can do to study a gene, and that's how I started or got connected from the beginning with Arabidopsis. So as I mentioned, so after that, so I stayed, so I worked, well, let's say in total almost 10 years in Wageningen. So I finished the PhD. So I started the PhD, the reason was actually that I had to guide a lot of students, PhD students that came to the lab, and guide them, explain them protocols, experiments, help them. And I was seeing they were going to nice congresses and publications. And that actually at a certain moment was one of the things like, actually, that's quite nice, and maybe I should do this myself. I think Nayelli also helped me in convincing this, that why not, no? So I did this, and so the idea was also to stay actually at the bench, because I liked really doing the experiments. And in a way I knew, so doing a PhD later on, for instance, you have to give presentations. In the beginning, it was really something that I didn't like. No, and you go from the bench, you go to an office, you go to the desk, no, so things really change. But in the end, it was really, and it is really, it was a good decision to make, and I really enjoy what I am doing in this moment. So doing the PhD, I started, I worked in the lab of Gerco Angenent. That was actually a lab on petunia, flower development. And actually with me being there, starting there, more Arabidopsis was introduced. And so I was involved in many EU projects. So one of them was the Regia project, which was a very ambitious project, where the idea was to clone all the transcription factors of Arabidopsis and functionally analyze them. Of course, that was a task maybe a bit too ambitious, but I think it was really a great project. So there were like more than 25 labs involved, European labs. So that also made it very easy to know people from other labs, make a scientific network, let's say. So that really helped me from that time, and still I have the, let's say, the benefits from that now. So many people that I met at that time are still good collaborators at this moment. So when I submitted my PhD thesis, so normally you have to wait a few months that people, let's say, external committee reads it. And then actually, so we came to Mexico in 2006. So already was with happily married. So I'll just mention that in a minute. So we went to the UNAM, the National University of Mexico in Mexico City, in the lab of Elena Alvarez-Buya, to also see if we could collaborate and start work on gene networks, using also a lot of protein-protein interaction data, what I was doing at that time. So I think that was very interesting, illustrative, how it is to live in a really big city. So there I got my conclusion that I did not want to live in a really big city. And that was also the time that we came to Irapuato to CINVESTAV, and I gave a presentation and saw how the institute was here. And we got to know that the plan was to make a new institute, which is Uga Langgebio of CINVESTAV, and that probably soon they were going to look for researchers. So I stayed more or less 1 year with Gerco as a junior researcher, because I finished the PhD and I still had my permanent contract, so I could just happily stay in the Netherlands. Let's say maybe the good or the bad thing is how you look to it, because it was a perfect place, and I think a lot at the beginning of my career in being there with all the colleagues and in Gerco, of course. But it was, let's say, the structure was not that you could easily start your own research, and that's why it became attractive to start to look around. And so I applied to start a lab in Mexico, and they called me and offered me the job in 2007. So we moved to Mexico. So I looked yesterday for some old pictures. I found some. So from the beginning, I started with Arabidopsis and I had to learn how to transform Arabidopsis, try to optimize it. And so this is also how I met Nayelli, so my wife now. So she was in the same corridor, but in the lab of Andy Pereira. And let's say the bosses talked and they said, there's this guy that transform Arabidopsis, maybe you should look how he does it. And so I got a task to teach Nayelli how to transform Arabidopsis. And to thank me, she invited me to eat quesadillas, and after that we ate many quesadillas more. So in 2000, I also found this. So it was it's in Dutch, it's in let's say from the institute, a newspaper that they published. So in 2000 we married, and like that our collaboration that started with transforming Arabidopsis ended in a very good marriage, and now still in my favorite collaborator. So some more than about the question. So my career path. So a few years ago, this is in 2019, I wrote actually the story how I came to Mexico. And there's a QR code if you want to find this article. And also it has more, let's say, my vision in how to search for a good lab and good collaborators and how to do your science. And so maybe this is interesting to read. And so why did I study plant sciences? So this is a very old picture. So I had my small old built greenhouse. So I always loved cacti, and I also grew a lot of them before ever have been to Mexico. And so when I met Nayelli, it was, of course, very interesting for me to visit Mexico. So in '98, it was the first time we went, and now I have a garden with cactus in my back garden. So this is why there's this title, and I think you can read more in detail my story also in this article. So what inspired me? So as kid, as I mentioned, I always loved plants. I think my parents, we always had gardens. There were always animals around. I guess this is why I loved plants. So this is a picture of the hand of Gerco. That was my PhD supervisor. So this, I guess, I took this picture like 25 years ago, and I still use this image in almost all my presentations. And so studying flower development. So so when I was more starting the PhD, I was reading all these papers, and there were all these big names that were studying flower development. They were cloning genes. So in the beginning, this was really, let's say, scientific heroes. Like, wow, they do really great science. Elliot Meyerowitz, Marty Janowski, Enrique Cohen, David Smythe, John Bowman. In the genomics, more Joe Ecker, Detlef Weigel, Philip Benfey, of course. In that moment, you have an idea, you feel very small, and you think, wow, these big guys, how do they do this? And during the career, you grow, you start to go to congresses, and then it's really nice actually to meet all these people, and you see, actually, they are just people, they are very nice people, and you can just talk with them. So this was, I think, very eye-opening and a very inspiration. And maybe some local Dutch heroes, of course, Martin Koornief started important in the Arabidopsis field. another person, Ben Scheers, and then developmental biology. And then actually, so my first Arabidopsis conference was in 2003. So still quite young, going to the US for the first time, very far away. So I presented this work, and I still remember I had my poster, and then I remember all the people coming, and then various very nice people, but friends from the lab of Martin Janowski. But I remember I was like scared, very nervous, like, oh, they're going to ask me things. Maybe I cannot answer the questions now. So in the beginning, I think probably many will feel like insecure, quite scared. But well, this is one of the many groups conference where I went, and still they are really always very great. So some the challenges that I have faced. So I have to say, my career went quite smoothly. So I think it was a bit different pathway doing a PhD by having a permanent contract and being in a place where I already had worked quite some years, being with very nice people. So this went quite well. Coming to Mexico, I guess there were more challenges. The first one was learn Spanish. And maybe I knew, I don't know, 10 words of Spanish coming here to Mexico. So this was, of course, very big challenge. So we come on the next slide to that. So my big help was my wife, Nayelli. We started together the lab. This made all the difference. Other things, adapt to different life and working habits, do more planning, stay patient. Things in Mexico are not so fast, no? So I was used to being in Europe, in the Netherlands, things maybe in 1 or 2 days, things arrive, or you have your oligos or your sequence back. So this is quite different here. Deal with high level of bureaucracy, which is quite high. You manage, but you have to get used to it. And another thing is do science with very little money, actually. From one side, I think we can, as Mexicans, be very proud that with very little money, we can do still quite nice things, nice science. So maybe you have to be more creative, think about what you really want to do. But this can also be an advantage. But of course, it's a big challenge. And in the last years, things became quite worse. When I just started in Mexico, things were quite different, and grants were approved normally, and now things it's more hard. So who helped you along the way? So first, well, in the Netherlands, my ex-boss, Gerrit Wankenent, and let's say, and all the colleagues, for instance, Richard Eming. So we did many things together, Plant Research International. So my wife, my favorite collaborator, Nayelli March. So we started the lab together. So I think more or less 4 years we were together in the lab. And after that, Nayelli started her own lab in, let's say, the unit next door. So there are 2 units here in Irapuato of CINVESTAV. CINVESTAV itself, so also Stewart mentioned, so Luisa Estrella, who hired me, who made the institute. So very grateful to that. It was a very good start, a very good opportunity to come here in Mexico, and it made also quite, it made starting the lab more easy. I have to thank my international collaborators, and also many of that come back or are back from the Netherlands and from these different EU projects. So also that we have very nice collaborations and friends at the end. Now, and also various Mexican colleagues and also friends, they help me doing the way here being in Mexico. So the lessons that I have learned, so I guess many, but so I wrote here, maybe a bit different than that we heard also in the other talks maybe. So the first thing is coming from the Netherlands. So Mexico has many talented students. They can come from everywhere. Let's say in Mexico, it's not only from, let's say, Mexico City, and many times maybe it's from the small villages, small towns. We get very talented students. So I'm very happy with that, and that helped my lab also a lot. One of the talented students is Vincent that we have here. The facilities that we have at CINVESTAV, actually, they're really good. Of course, people like to complain that they do in each country. I see in Mexico, people like really to complain, but we have it actually very good. There are very different conditions in Mexico, of course, that they acknowledge. But CINVESTAV is really great. So we got many visitors and many visitors of the European labs, and they are all very impressed when they come to Mexico, what they see here. I think it's very important to make time to think about what you really want in life and in science. No? So let's say it's really a two thing in life itself, but also in science. If you want a lab, do you want to do a PhD? Yes or no? It's really worthwhile to think about this. So being in the lab, before when I was in the lab, believe in experiments that you want to do. So otherwise, don't do it. Use the opportunities that come along your path. So I had various of them and I took the opportunities. Treat people how you want to be treated. I think it's a very important thing. Another one is choose your collaborators wisely. Do not lose too much time with politics. You can lose a lot of time in that, so take care. And also what Nayelli mentioned, celebrate more. I think this we learned very late on in our career. And so the moment we came so much in the work, and it was like next paper after the other and not really celebrating. So I think it's an important thing, and I think that we are still learning. So with that, well, is my story. So thank you to all the lab members. I didn't put the names, also not to forget names. So there are many names, many people. So thank you all to that. Everyone contributed to the science that we did. Thanks to all the collaborators and thanks to all the colleagues. Here we have some old lab pictures and new lab pictures. I think we had many nice parties. I think that they know very well to do in Mexico. So thank you all for that. And that's my story. Thank you.
Thank you, Stefan. It was a really, really good presentation. Thank you so much for agreeing and accepting this invitation. We actually don't have that much time for questions, but I would like to collapse these two last that I got. I want to ask Nayeli, Stefan, and Stuart if you could decide, I mean, if you can go back in time and if you were asked to move to Mexico and do Arabidopsis science again, would you do it again? I want to start with Stefan, that is the last that talked.
Yes.
Yes. What about Nayeli?
100% yes.
Thank you. Nayeli or Stuart?
Probably they cannot unmute themselves.
Oh.
Nayeli, maybe you can go first.
I think if it was in the past, yes. At this moment, it's actually quite hard. I'm hopeful that it will improve. But if it would have to be now exactly, I would have to think it more carefully.
Yeah, sounds fair. What about Stuart?
Maybe a little bit similar. I think if it's me in particular, I'm glad that I came here and that doing what I'm doing. It's very challenging now. I think it depends entirely. There's going to be a different answer for every person, depending on especially what your opportunities are and what you want to do with your life. But we have lots of good things here. But the less good things, unfortunately, have been growing. But yeah, there's still a lot of great things about being a scientist in Mexico, especially the students and just that. Yeah, as Stefan mentioned, in general, stuff is more fun here. Almost anything. So it is important to have fun in life.
I think that I have. I'm sorry.
No, maybe just to add to that. So I think the academic freedom that we have is really a super good thing here in Mexico. So this also makes things fun. You can really do what you like. That's why it's also important to think about what you want. No?
Yeah. Thank you. I think that I have time for one quick question, and would you say that the line that you started in your PhD was the line that determined your career path or your like destiny, we will say that the line that you are doing research now? Do you agree
If
with that?
if I it's I mean that's why you maybe, but maybe not. That's why you do a postdoc. You're actually a postdoc is not supposed to be just doing more of what you already did. That's not really the idea. The idea is really to learn something new. So you can kind of branch out a little bit, or you could do something totally different. Obviously, that's more challenging, but your PhD doesn't necessarily determine what you're going to do later. That's my opinion.
Thank you. Stefan, Nayeli, you want to add something to that?
And Nayeli's microphone. And Nayeli can go first.
Yeah, you can go, Nayeli.
Thanks.
I agree with Stuart. So in my case, what you learn in your PhD, well, when I learned in my PhD has really helped me a lot with what I do now. But for example, I realized that it was not my dream to make mutant populations, And I found that I really love the mutants themselves, and I wanted to study them further, more than making new strategies to make mutants. Which I still find interesting, but now I'm doing less.
Stefan,
I think in my case, yes, it determined where I am now. First of all, deciding that I did a PhD, otherwise I was, I guess, not here. And then being in the lab where I studied flower development, so this was very focused on matchbox proteins and only matchbox proteins, which are very cool. So I moved away from that to, in a way, not really touch them, but just in general transcription factors. But I think I was or am, let's say, contaminated to find transcription factors very important. And then when I moved more to carpel and fruit development, and so yes, it determined where I am now in the end. Yeah.
Well, I think we are right on time. I want to thank a lot, Stefan, Nayeli, and Stuart for accepting this invitation and accepting to share their history in biology, in plant biology especially, because I mean we are like a Arabidopsis community that more than Arabidopsis, and since I mean Arabidopsis is like the main thing, but this is like focused like to all plants. And I would like to finish saying that yeah, Arabidopsis community is open to all countries, and you are more than welcome to join this. And I will say I really wanted to do this webinar with Mexican representation. I really wanted to do this webinar with people that were not necessarily from Mexico, that but they were doing science in Mexico, which is like really good, I would say. And I really want to thank you for accepting this. And I don't know if Joanna was has something else to say.
Oh, sure. I can just follow up. Thanks, everybody. Thanks also especially to Vincent for organizing and really proposing this great idea for the My Plant Biology story. This is the second edition. We had Jose Deneni, who did a really wonderful talk a couple years ago. He's at Stanford. And when we had a committee meeting about how we would do this next edition, the committee really loved Vincent's ideas of bringing in scientists from Mexico. And we thought it was, I personally thought it was a great mix because Stefan and Stewart weren't from Mexico, but they ended up there. So we thought that with Nayeli's perspective really could provide valuable insights, because so many of the scientists, it's an international endeavor, and many scientists move around the world. And so we felt that I felt that this was a really great way to do